Okay, we're going to look at Chapter 9, Campbell edition on cell respiration. When we look at this panda bear here eating, let's just break this down a little bit. Literally, light energy went into the plants to make organic molecules, as well as some oxygen. These are then broken down by the panda bear using oxygen inside its mitochondria to make ATP energy as well as heat. Carbon dioxide and water left those processes and refueled photosynthesis. So what I want you to get from this is that these two processes are linked: photosynthesis and cell respiration. Cell respiration is the breakdown or catabolic pathways that release energy by oxidizing organic fuels like glucose. So when we talk about oxidizing, we mean that oxygen is going to rip electrons off an organic fuel like glucose to make usable energy in the form of ATP. This takes place in eukaryotic cells like human cells. We're going to talk a little bit about bacteria a bit later on. Oxidation reduction, just let's remind ourselves, Leo goes Gur, loss of electrons, gain of electrons, an electron donor is a reducing agent, and an acceptor, electron acceptor, causes another compound to lose electrons, so it's an oxidizing agent. And energy is associated with the movement of electrons. So in cell respiration, we've seen this before. Glucose becomes oxidized, and that's its oxidized form there, and oxygen becomes reduced, energy is formed. Oxidation reduction reaction. Other energy contributors is NAD and FAD. Okay, when they gain electrons, they become reduced and they have more energy. And these are very important electron proton carriers, okay, in cell respiration. So the reactants like glucose, as they become products, low energy products like water and carbon dioxide, involve oxidation reduction reactions that produce ATP. Glucose, we know, high energy, soluble, and small. Cell respiration is going to break this glucose down into ATP. Now, the glucose, about 36% is used to make ATP, and about 64% is released as energy. So 36%, this is not 100% efficient. And the other 64% is lost as heat. That's why you heat up when you eat food. ATP, okay, it's used in cell processes. Some examples of this, if you remember from Science 10, ATP is used to pump molecules from an area of high concentration of low, or low concentration of high concentration. You're going to learn a special case of nerve transmission involves the sodium potassium pump in nerves. So it's important there. This bacteria is flagella, is fueled by ATP, okay? Or sperm cells. And we've learned about muscles before, okay? We've learned about the oars, and the ATP allows the oars to be released, the myosin oars to be released, okay? And that powers up a muscle cell. Okay, types of cell respiration. There's aerobic and anaerobic. So without oxygen, we have anaerobic and fermentation. Two types we're going to learn about later on. These two types right here. There's 4 stages of aerobic and respiration, and anaerobic fermentation will also use the first stage, glycolysis. So glycolysis, pyruvate, Krebs, and electron transport chain chemiosmosis. Occurs in the cytoplasm, matrix of mitochondria, matrix, inner membrane of the mitochondria. That's the location. So this is some diagrams from your textbook. Glycolysis in cytoplasm. It breaks down glucose, so that's the substrate at which ATP phosphorylation occurs. That's then going to enter the mitochondria for aerobic cell respiration. Inside the mitochondria, we have, so this would be stage 1. Stage 2 occurs here as well. It's pyruvate oxidation. Stage 3 is cytocitric acid cycle, also called the Krebs. And stage 4 is electron transport chain and chemiosmosis, and that uses oxygen as final electron acceptor. So we call that oxidative phosphorylation. Substrate phosphorylation uses a substrate, glucose, to make ATP. Here, we're using the strongest electron acceptor, oxygen. Now, most of the ATP is made here, 32 to 34 ATP. Here, another 2, another 2. So we add that up and we get 32 plus 36 to 38 ATP are made for one glucose molecule that enters the cell. So we're going to cover the first part here, glycolysis. We're going to take glucose, we're going to cut it into 2 pyruvic acid molecules. This does not require oxygen, and we only make 2 ATPs and a little bit of NADH. NAD is reduced. Here's another great analysis of it. Glycolysis takes place in the cytoplasm. It can take place in bacteria, plants, animals. Glucose gets converted into 2 3-carbon compounds called pyruvate. 2 ATP are used to bring the glucose in, but 4 are made. So we go 4 minus 2 means that we have a net production of 2 ATP. And a little bit of NADH is produced. And this is just another way of looking at 2 pyruvates. 4 are made, 2 are used, a little bit of NADH. This is a nice little overview you can take a look at as well. Aerobic cell respiration takes place in the mitochondria. Glucose when it enters into the mitochondria, it gets converted into acetyl-CoA. And we're going to look at that next. Well, let's just remind ourselves of the mitochondria. This is an electron micrograph of a mitochondria. And it kind of looks like a footprint on the moon, I guess. Here are the parts. Know them. The inner part is the matrix. The inner membrane has these really increased surface area foldings called cristae. So they're part of the inner membrane. They're very, very important because that's where the electron transport chain occurs. And there's the outer membrane. That means that in between the inner and outer is an intermembrane. And the intramembrane, there it is, right here. So we've got an outer, an inner membrane, as well as an intermembrane, and a matrix. Well, pyruvate oxidation. Before the Krebs cycle begins, pyruvate is changed. And this occurs in the matrix. A carbon dioxide is lost, and the coenzyme A combines with the pyruvate to the molecule where the acetate molecule, the 2-carbon molecule, to form acetyl-CoA. And a little bit of NADH is formed that'll go to the electron transport chain. Here's my pyruvate, 3-carbon compound. A carbon dioxide is lost. NAD is reduced. Coenzyme A combines to form acetyl-CoA. Where does this take place? In the matrix. CoA, acetyl-CoA, carbon dioxide. Okay, we are going to look at the Krebs cycle in the next lesson.